Thank you. Thank you for coming for the last talk of that Proceedings track. So let's start about the linear effects models. Can you hear me? Yeah? Can you hear me now? Okay, good. So the usual way we study signaling pathways is by the means of perturbations. So we use an experiment which targets a component in a pathway, and by doing that, we block the signal flow in the pathway, so via the pathway, and via some intermediate layer of control of effects via the pathway components. So that control can be, for example, gene regulation. And then we observe effects of that perturbations, like, for example, gene expression or protein concentration or whatever other phenotype that you wish to measure. Okay, so from these perturbation effects, we wish to infer two unknowns, the structure of the signaling pathway and the way the pathway regulates the effects. So if you look at it, at this picture, you immediately realize that inference in such a multi-layered system is very hard. And yes, so we perturb one hidden layer, which is not observed on the top. Then this goes via another hidden layer, and only then we make observations on the endpoint of the entire system. Moreover, the components of the pathway are all interconnected. And that means if you perturb here, this perturbation propagates downstream to other components of the pathway. Finally, the intermediate layer is non-trivial. Okay, so you can assume that each of the perturbed components has its own contribution, and they jointly make what you observe as an effect. So to nevertheless make inference in this system, we have to somehow make simplifying assumptions. So there are different groups of modeling approaches depending on which type of assumptions they make. One group just strictly focuses on this first layer of the signaling pathway. So they either do this by direct measurements of the states of the signaling genes, which is rare, or they approximate them with gene expression, which is a crude approximation. Other types of models actually are even more optimistic, so they assume that the layer one is known, its model is given, and they observe layer 3 of the effects, and they only model the intermediate layer of control. Finally, there are models like the nested effects models, which do account explicitly for the signaling, for the intermediate layer, and for the observed effects. But they oversimplify this intermediate control. So in these nested effects models, you assume that each effect is only regulated via a single gene in the pathway. Okay? So in this work that I'm presenting now, we try to make a bit less simplifying assumptions. So we propose what we call a linear effects models, abbreviated LEM. And so we assume that the experiments target genes in the pathway, and the pathway is represented as a graph. So the nodes stand for the genes, and the edges in the graph show how perturbations propagate in the pathway. Each gene G is also associated with a contribution to the effect, which we denote beta G. And now the most important assumption of the model is that the effect that you see is a sum of the contributions of the genes that are perturbed in the pathway, either directly or by propagation in this pathway. Okay, so to understand this, first remember that graph, okay, this example graph, and then focus on panel C. So in panel C, on the x-axis, you see the different possible experiments that can be done on that graph. So an experiment is a set of genes, is represented as a set of genes that are directly targeted by that experiment. So consider an experiment where you only directly target gene 1 in this pathway. So in this, when we directly target gene 1, there are no other genes in the pathway to which that perturbation propagates. So you observe as an effect only the contribution of that gene, beta 1, denoted here. If you then have an experiment that directly targets gene 2, which is on the top of the pathway, then this perturbation propagates down to genes 1 and 3. So as an effect, all of them are perturbed in that pathway. So what we observe, according to the assumption of our model, is a sum, so that's here, the sum of contributions of beta 1, beta 2, and beta 3, okay, and so on. So I just wanted to say that it's a very nice coincidence that the name of our model, LEM, is also the last name of my favorite science fiction writer, Stanisław Lem. Okay, but so coming back to the LEMs, you cannot see this somehow, right? It's like shifted to the left. I'm sorry. So it says LEMs a bit more formally. Can you maybe move it? Oh, okay. Maybe I do like that. No. Yes, it's better? Okay, so so a bit more formally. So we have E, a set of experiments that that denotes a set of experiments that target genes. The genes are denoted from 1 to n. And we have a G, which stands for the pathway graph with edges that show this propagation in the pathway. And we assume that G's are transitively closed. And now when you have E and G, we define what we call a binary perturbation states matrix. So that matrix has rows for experiments and columns for the genes, and it just says which gene is either directly or indirectly perturbed in which experiment. So formally, this means that the entry SEG for an experiment E and gene G is 1 if experiment E directly targets G, or it's 1 whenever any of the parents of G in the graph G is has value 1. Okay, so it's a disjunction of the values of the parents. We also have parameters which are a vector of parameters beta, which stands for the contributions of the genes to the observed perturbation effects. And finally, we have y, which stands for the single observed variable of the perturbation effects. So now this linear assumption of the model is encoded here. So we say that YE, so the measurement under experiment E, is given by the linear combination of the contributions of the genes weighted by their perturbation state, plus some Gaussian noise. So that can be, is also represented by SE transpose times beta, where SE transpose is just a row in the S matrix that corresponds to experiment E, times this plus noise. So this should remind you this formula of a linear regression. And that's exactly what we use, but in a Bayesian version of it, because it's more general and it allows you to do more things. So because it's Bayesian, we are dealing now with a marginal likelihood of our model for given experiments and the given graph G. We can compute this S matrix, the perturbation states matrix, and now the likelihood, so the log likelihood looks like this. So we have here, these two terms together, this is the posterior. So here is the standard likelihood that you have in the linear regression. So here we assume that y is Gaussian distributed with the mean S times beta, as I told you, and then we have invariance matrix, so we have C to the -1 times identity. So the C is the precision parameter, which is just inverse variance. And here we have so we have assume a prior on the beta parameters. It's a zero mean Gaussian prior with precision B. And here we integrate the betas out. And then we also have an additional term which allows you to set priors also on the structures of the graphs. So all of this is actually the standard procedure that was developed for the Bayesian linear regression. So for example, the way to estimate parameters in maximum likelihood parameters for that is all defined in textbooks. So you can read that up in our paper, or for example, in the pattern matching book by Bishop. So we apply that iterative procedure of estimating the parameters, and then to solve this first problem of inference of the parameters, so this intermediate controlled layer, that's our betas. So to do this, we just set the betas to the mean of their posterior distribution in that model. So now, how to solve the second problem of inference of the pathway graph when we are only given experiments E and what we observe in these experiments. So we have to somehow search the space of possible graphs. And when we do that, each time we have a new candidate graph, we evaluate it with its marginal likelihood that I just introduced you on the previous slide. And to search the space, we use exactly the same procedures as were developed for nested effects models, because the pathway component is the same in our model. So we either exhaustively search over all small transitively closed graphs, or we use a greedy procedure like adding or removing edges. So okay, so now once we have introduced these models, some interesting questions arose. One of them was about identifiability. So let me just remind you that the models which are identifiable, for them, if you have two different models, so for us this means different pathway graph, different parameters, they cannot have the same likelihood. Then they are identifiable. So intuitively you can think, okay, we need to have enough data from which we infer the models for them to be identifiable. So the question is, how many experiments do we have to do for the LEM models to be identifiable? So we studied that problem in the paper, and so you have these lemmas here. They tell you that it's not enough for identifiability to have only single nodes perturbed. It's not enough to have all pairs of nodes perturbed. You get identifiability of LEMs if you perturb both perturbations of all single nodes and all double nodes. Okay, that means n+1 choose 2 experiments. So what's the conclusion from this? There's no free lunch. So that means, I told you, we want to have more expressible models, we want to make less simplifying assumptions, and then we have to pay for a price for it with making more experiments. So we wanted to study the performance of our model in a controlled setting where we know the true models and the true parameters. So I'm going to show you how well we can learn the pathway structure using simulated data. So here on each plot, on the x-axis, are the numbers of repeated experiments, so technical repeats, simulated technical repeats. And colors show increasing levels of noise from orange to blue. So blue is very large noise. And the first row stands for when we do exhaustive search, and the second row when we do greedy search. So here I show the fraction of pathways that we learned perfect. So means we learned all the edges and we didn't add any false edges. And that's actually, we learned perfectly the pathways for low noise. Also, if you look at sensitivity and specificity of learning the edges, the median, regardless of the noise, the median is 1. But if we go to greedy search, then this fraction of learned perfect goes down by 40% for all levels of noise. However, if you again look at sensitivity and specificity, it stays on a very high level. It doesn't decrease less. It doesn't go less than 0.8 for the worst case. So then we checked what will happen if we violate this model, the linear model assumption. So this shades of gray show increasing number of pairs, which contribute a pairwise interaction term to the observed data, in the simulated data. So So that's increasing violation to the model assumptions, okay? So if you do that, then this fraction of learned perfect drops dramatically to like 0.1. But the sensitivity and specificity, they actually stay on a very reasonable level, so they don't go lower than 0.7. We also applied it to a very small case on real data, so we learned the direction of flow between PBS2 and HOG1, and we learned which genes are regulated by the single HOG1 or both of them or PBS2. And with this, I want to thank you for your attention, and I want to acknowledge my co-author, Nicole Brennwinkel, travel funding by the Warsaw Center of Mathematics and Computer Science, and I want to direct you to our R package. So thank you for listening, and I'm happy to take questions. Thanks.